Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. I'm very happy to be back with you today to discuss, or I should say further discuss, the enzyme in question, which is pyruvate kinase, which is one of the last steps actually of our glycolysis journey. Now we have a couple other steps to hit because we will be hitting the kind of pathologic Warburg substrate level phosphorylation metabolism, where lactic acid is involved and lactic acid transporters and stuff like that. So we have a couple more steps we have to hit, but I had to unfortunately postpone this meeting because I've been working as a hospitalist the last week, and as much as I wanted to share on pyruvate kinase, finish up sharing on pyruvate kinase, I'm really excited that I kind of actually did postpone it, because I did add a few slides to our discussion today, and I think that they really will supplement and enhance what we're doing. Welcome, Paul. Welcome, Cancer Outside the Box. Happy to have you guys here. Let's get started. What do you say? Okay, so we left off on this slide, and we were kind of talking about all the ways, or some of the ways, that pyruvate kinase, not only like what its main job is, but also like some of the things that upregulate it and promote it. And we talked about things like insulin, and how basically insulin could be driving the upregulation and activation of pyruvate kinase. And that would be a major benefit. As a matter of fact, I was very surprised to read that I was reading a paper on ketosis for cancer by Seifried and his group. And they believe that the major mechanism behind why a ketogenic diet was actually good for cancer was not primarily because of the low glucose. It was actually because of the low insulin and because of the ketones that were present. And the low glucose was actually kind of a tertiary or kind of less important role than those two, which I was very surprised at the first time I read. But when you realize that insulin is such a strong growth factor, and when insulin is elevated, it can upregulate multiple steps of the Warburg metabolism and damage mitochondria and stuff like that, it makes a lot of sense. So this paper was titled Pyruvate Kinase M2 and Cancer, the Role of PKM2 in Promoting Tumor Genesis. And it was published in March of 2020. And it says pyruvate kinase plays a pivotal role in regulating cell metabolism. The final and rate-limiting step of glycolysis is the pyruvate of phosphoenolpyruvate, PEP, to pyruvate, which is catalyzed by pyruvate kinase. So this part of the story we've actually talked about, that it's the last step in glycolysis in kind of the normal physiologic conditions. Normally, pyruvate would get put into the mitochondria and then further oxidized through the TCA cycle and the electron transport chain. Remember, we're talking about cancer metabolism, so it's a different situation. There are 4 isomeric tissue-specific forms of pyruvate kinase found in mammals: PKL, PKR, PKM1, and PKM2. PKM1 and PKM2 are formed by a single mRNA transcript of PKM gene by splicing, or alternative splicing. The oligomer PKM2 exists in high-activity tetramer and low-activity dimer forms. The dimer PKM2 regulates the rate-limiting step of glycolysis that shifts the glucose metabolism from the normal respiratory chain to lactate production in tumor cells. Besides its role as a metabolic regulator, it also acts as a protein kinase, which contributes to tumor genesis. This review is focused on the metabolic role of pyruvate kinase M2 in normal cells versus cancerous cells, and the regulation at the transcriptional level. So what are these dimers and tetramers? What are they talking about here? So A dimer is a protein that is connected to another protein. So there's two proteins. That's called a dimer, two proteins. And if those proteins form four proteins, that's called a tetramer. So the way it should be happening in kind of normal physiology would be that this PKM2 would form this four protein structure, this tetramer, and that would lead kind of like normal physiology to happen. But when PKM2 is in its dimer form, or it's kind of like more smaller form, it's what's going to be used mostly in cancer. And that means that the pyruvate is going to ultimately end up in lactate. And that's kind of the difference between the normal and the healthy cells. I also said like PKL, PKR, PKM2, and PKM1. What is that talking about? Well, PKL stands for PK liver. PKR, I believe stands for PK red blood cells. And then PKM1 and M2 are PK muscle 1 and 2. And you can see that there's a variety of different types of PKs in different types of tissue. So of course, PKL you would think would be in the liver. So it says hepatocytes, but then it's also in the intestines, pancreas, kidney. PKRs and erythrocytes, red blood cells. And then M would be heart muscle, skeletal muscle, smooth muscle, brain, adrenal medulla, mature sperm, and then PKM2, PKM1. And PKM2 is in tumor cells, embryonic tissue, testes, ovaries, adrenal cortex, pancreatic islet cells, thymus, kidney distal tubules, and leukocytes. And here's like the larger tetramer for protein structure, and then here's the dimer portion of, and basically like what we're going to see is that these two different forms will, how do I say, will kind of like be back and forth switching, and that will help with redox homeostasis. But I'm going to show that in a picture format so that can make more sense. So PKM2 and cancer metastases. So it is well known that many cancer cells grow under anaerobic conditions without oxygen. However, unlike PKM1 or the tetramer of PKM2, the dimer PKM2 synthesizes minimal, if any, ATP during the PEP to pyruvate conversion. Thus, the net ATP gain for glycolysis is 4 minus 2 minus 2 equals 0. And it's already known that biochemical pathways are reprogrammed in many cancer cells, but it remains unclear how these cells generate sufficient energy with abnormal mitochondria. Tumor cells under hypoxic conditions, when the dimer PKM2 is active and OXPHOS is suppressed, get energy from mitochondrial substrate level phosphorylation. And that's that. In fact, I've watched an interview recently with Dr. Duraj, and of course I've read the most recent CFRE paper that was just published, which I'm really excited about. Sharing with you. I think that I'm probably going to do it in a video format instead of a live. But my point is that they're using the term substrate level phosphorylation. They're saying that is a more accurate way of saying basically the Warburg effect or aerobic glycolysis. So I'm seeing this in other papers as well. Mitochondrial substrate level phosphorylation. That's basically Warburg metabolism. The literature suggests that when oxidative phosphorylation is impaired in tumor cells, the succinyl-CoA ligase reaction in the tricarboxylic acid cycle, or TCA cycle, can substantiate sufficient ATP through mitochondrial substrate level phosphorylation for the growth of cancer cells. Production of high energy phosphates would be supported by glutaminolysis, or glutamine metabolism, through the subsequent conversion of glutamine to glutamate and then alpha-ketoglutarate, which is further converted to succinyl-CoA, then succinate. It is well documented that tumors require large amounts of glutamine for survival and growth, and that glutamine is a major source of energy for cancer cells. Glutamine not only provides nitrogen for the synthesis of nucleotides, or DNA, RNA, and NEAAs, nucleotide and non-essential amino acids, but also provides alpha-ketoglutarate to serve as a precursor for ATP synthesis through substrate level phosphorylation at the TCA cycle. So, and then it later says that lipid biosynthesis, in the final and rate limiting step of glycolysis, the expression of PKM2 is upregulated in lung, breast, cervical, kidney or renal cell carcinoma, bladder cancer, papillary thyroid cancer, colon, and prostate cancer, and very likely all cancers, considering it's a hallmark of cancer. PKM2 protein switches between a very highly active tetrameric form and a low activity dimeric form in healthy tissue. However, it has distinct properties when acting as a dimer in tumor tissues. Numerous studies revealed that dimeric, or the smaller 2-protein version of PKM2, has reduced catalytic activity, which participates in macromolecular biosynthesis, thus generating the materials needed for rapid cell proliferation. Apart from this role as pyruvate kinase, it functions as a protein kinase and cell transcriptional cofactor of many genes associated with tumor cell growth, metastases, and cell death. And the next part I really wanted to show from this paper was excellent. It was a really great highlight of how PKM2 is different for cancer cells and kind of like how it functions for its metabolic properties, but also for its growth, metabolism, redox homeostasis properties, which I think is going to be important for us to know. But it also talks about how it's triggered and what it does. So we see here that insulin-like growth factor, and we subsequently, or previously saw that insulin signaling also, and it does say IGF-1 here too. But basically, like, these growth signals are increasing the expression of PKM2, which is going to lead to tumor angiogenesis and tumor growth. You can see here that IGF-1 directly will interact with HIF and PKM2, which is why they're saying it's not just an enzyme that takes place in the cytosol that converts PEP to pyruvate. It also will go to the nucleus and actually signal to participate further, above and beyond that of just a normal enzyme for metabolism. Moreover, enhanced expression of PKM2 is associated with the worst overall survival, OS. Numerous studies have reported that PKM2 is highly expressed by regulating the expression of this NEK2 in multiple myeloma, indicating that it could serve as a prognostic marker. In multiple myeloma patients, poor prognosis is associated with higher levels of PKM2 gene expression. High tumor cell metabolic activity and proliferative capacity are reflected by PKM2, so it serves as a non-organ specific molecular marker. Additionally, some PKM2 is also related to neighboring tissues of necrosis of cancer cells and tumor cell renewal, which can be used in an early diagnosis of cancer. The PKM2 level in circulating blood of patients may serve as a prognostic or a potential diagnostic marker in various carcinomas. From my understanding, I've never tried to order it, at Quest, but I would highly doubt that this is available for clinicians to order. I definitely don't have it at the hospital. But what it's basically saying is because it's so highly associated with, Warburg metabolism and cancer kind of aberrant growth, that some of it will spill out into the bloodstream when there's cancer necrosis or uncontrolled cell death. And so because there's going to be some high level of it, higher than normal level circulating in the bloodstream, it's possible that in the future, definitely not now, that it could be used as a way to detect whether you have cancer or if your cancer is doing very well or not doing well at all. So we're seeing that, and you'll see this later with other markers, right? Like, for example, lactate dehydrogenase. That's something that we can easily check. And lactate dehydrogenase is what converts that pyruvate that's being converted by pyruvate kinase to lactate. And LDH is something we can check very easily. And I have been actually checking on a lot of people, cancer patients in particular. And I find that it's basically showing how glycolytic is your tumor. And obviously, most cancers are glycolytic. So it's really the higher, the worse it is. Tired of looking for name. Thank you for your support. Fernando says, Doctor, thanks for your work. What can you think about nicotine as off-label medication for cancer treatment? So I'll be honest with you. I have done my own little deep dive into nicotine because of work that one of my mentors, Dr. Cruz, for a while was kind of like promoting its benefits. And then, of course, like I'm not sure if you're familiar with the folks over at Transcriptions. They have a troche called blue cannatine, and that blue cannatine has nicotine as well as methylene blue. And I've tried it, and there's a lot of like kind of like neuroscience type folks or kind of biohacker types that are like trying to get on board with nicotine. The issue that I found pretty quickly when looking through the medical literature is that nicotine is so demonized because of smoking and vaping and stuff like that, that I could not really find a lot of like helpful studies. It was very like negative studies. So I'd have to look further, but I would say that it's probably not very high on my list. I try to like put things high on my list that are like, that have either survival benefit. I've been really focusing on things that have a survival benefit recently, and then things that dovetail or make sense that could dovetail into metabolic therapy. And I'm not really sure how nicotine would dovetail in the metabolic therapy this time. Does it mean that it won't? Maybe it will. I had I wouldn't say similar, but I've had other things, I should say, that I have been maybe somewhat bearish on. The best example I have right now is ivermectin. And then, I did my deep dives or multiple deep dives, and I found that, wow, actually this is great. So I think that the same could be possible for nicotine. It's just that, again, when I did my deep dive looking for its effects on, improving mitochondrial function, I was left, not very excited about it, and a little bit, I just kind of put on the shelf and moved on to things that had better studies and better literature. It just doesn't mean it doesn't mean that it's not possible. I haven't seen it yet. Which reminds me, speaking of things that have a survival benefit, so I have completely like revamped my metabolic therapy course, and I'm going to be presenting it, 24th through the 27th, every day, 90 minutes. It's going to be hopefully a really good time. Every time I've done it before has been a great time. Obviously no one wants to be there, because if you're there, you have someone you love or you have cancer, but we all generally have a pretty good time presenting the information and, kind of sharing back and forth. And someone took a half of a blue cannatine this morning. Good for you. Yeah. I like blue cannatine. I actually would stack it with like a full dose of methylene blue, like that I had prescribed, or not prescribed, but I had to get from a compounding pharmacy, because there's very, very small amounts of methylene blue, and the methylene blue component, it's like a mg per kg dosing. So I wanted to try to stack it with the other one, but I did like it. Anyway, so my point is that, I've really revamped the course so that it has a much different feel to it than it did even like a couple of months ago, because number one, I'm exponentially learning, and I want to make sure that what I'm learning is in this course, not only like inhibitors of these things like pyruvate kinase, lactate dehydrogenase, hexokinase, et cetera. That list has like expanded exponentially, but also just like other things that are pretty important that I think a person can do and give themselves a better chance, with like clear survival benefits in the medical literature. Those things have been added. And then a couple other things that I've added to the metabolic therapy course, which you may have seen before, you may have not. I've really plussed up on the mechanisms, but also the benefits of the repurposed medications. And I've added to that, in particular, the membendazole, fenbendazole, ivermectin, statins, other things, aspirin, of course. Those have all been kind of added with the respective kind of new insights that I've been able to gain, doing these lives and doing these videos on these respective topics. And also have done deep dives into, whether or not they have survival benefit, which many of those do. And then lastly, I would say that another way that I've changed the course is that I've got a lot more information now about how to improve your GKI. That's probably one of the more difficult things with metabolic therapy is getting your glucose ketone index in the right kind of like space so you can, really dive into the other things and use the other things effectively for metabolic therapy. So those are some ways that I've, improved the course, and I think it's going to be a really good course. So if anybody's needing like really a lot of guidance, kind of like with getting started with metabolic therapy, that would be a good way to do it. All right, moving on. So protein kinase function of pyruvate kinase M2 in cancer. So now we're going to be looking at kind of the non-metabolic functions here and how important they can be. So this was published in October of 2020. And it says, in addition to pyruvate kinase activity involving the metabolic pathways, what we've talked about, converting this glucose to PEP, pyruvate enol phosphate, to pyruvate, this is the metabolic functions. We're going to be looking at the non-metabolic functions, okay? And it says here that pyruvate PKM2 has been shown to be translocated to the nucleus or traveled to the nucleus, right? And where it serves as a protein kinase to phosphorylate various protein targets and contribute to multiple physiopathological or pathological states, disease states. And the first thing it says here is that evidence supports the potential role of PKM2 in tumor genesis, actually creating the cancer itself. As an embryonic isoform, PKM2 is reactivated in tumors and overexpressed in multiple cancer types. The alteration of PKM2 activity is related to cellular proliferation and tumor growth. It later in the paper says that independent of its pyruvate kinase activity, PKM2 exerts a non-metabolic function such as pyruvate kinase activity and transcriptional coactivation. PKM2 has been observed to transfer to the nucleus, phosphorylate protein histones, and activate gene transcription, acting non-metabolic functions in cancer cells. For example, epidermal growth factor receptor signaling promotes transfer of PKM2 to the nucleus, and then the nuclear PKM2 binds to phosphorylated tyrosine 333 of beta-catenin. The complex of PKM2 beta-catenin is recruited to the promoter of the CD&D1, which encodes for the cyclin D1, leading to cyclin D1 expression and consequently promoting tumor development and cell proliferation. Evidence supports that a non-metabolic function of PKM2 and EGF-induced tumor genesis, EGFR, excuse me. So what is this functionally saying? I'm gonna get to this in a second. So what we're seeing here is that when epidermal growth factor receptor is activated by epidermal growth factor, it will basically signal to PKM2, hey, go to the nucleus and go turn on gene expression, right? So now we're looking at HSP90, PAK2, which is PAK2 is important for setting up the Warburg metabolism and enhanced glucose utilization. It's also interacting with the beta-catenin, like it says here. So now we have upregulation of GLUT1, lactate dehydrogenase, et cetera, and other parts of Warburg metabolism. It's involved with c-Myc. So we can see here that epidermal growth factor is using PKM2 not only to make, to metabolize more glucose. That's one of the things that it does. It upregulates glucose metabolism, aka it upregulates the Warburg effect. So it'll make more PKM2, what I was saying, and it'll allow for more glucose to be metabolized. But it has non-metabolic activity as well, as it acts as a transcription factor. It'll actually go and signal in the nucleus with other factors to help enhance cancer metastases and invasion to other tissues, et cetera. And that's what it's kind of talking about in this last piece here. It says the non-metabolic function of PKM2 contribute to the multiple process of tumor pathology. And in addition to tumor development, PKM2 is involved in the regulation of the Warburg effect, cancer metastases, epithelial mesenchymal transition, EMT, gene expression, mitosis, cellular proliferation, apoptosis, DNA damage response, et cetera, et cetera, et cetera. So it's more than just converting one boxcar to the next boxcar, right? It's more than that. It does more, and it's self-fulfilling. It's kind of a vicious cycle because when PKM2 is activated by growth factors, it will go up and upregulate itself. So it's like a vicious snowball. So then more of it's available. to be converting, glucose to pyruvate. And then on top of that, it's going to have more signaling capacity. So it's just, as you can see, or I hope that you're seeing, cancer is just a bunch of vicious cycles that have to be broken. And really the easiest way to break those cycles is to stop the cancer cell's ability to be able to metabolize glucose and glutamine, because ultimately what is happening here, pyruvate kinase is being phosphorylated. That requires ATP. If there's no ATP, these signal transduction pathways can't happen. You can't have cell growth and metabolism without ATP. So therefore, ultimately, although all these growth factors like EGFR and HER2 and androgen receptor and progesterone and estrogen receptors in different cancers, or KRAS and these things, these are all like targets that end up being a whack-a-mole situation. But what do they ultimately do? They ultimately upregulate Warburg metabolism, enhance glutaminolysis, because in order for these processes to actually function and do what they want to do, which is enhancing growth and leading to metastases and invasion of the tissues, they're going to have energy, and energy is required for life to sustain itself. And if you don't have the energy, then none of this stuff is going to happen. So therefore, the easiest way to break these vicious cycles is through, ketogenic metabolic therapy. This is another way of looking at it. We have EGFR, PKM2 is being phosphorylated. Phosphorylated PKM2 is going to the nucleus and then leading to gene transcription and ultimately, kind of like gene expression that leads to changes in the way the cell operates, more like a tumor cell, right? So another way that PKM2, aside from EGFR, aside from insulin-like growth factor, is mTOR. And I'm going to read the conclusion here, basically only because it gets a little bit long-winded even for me. But it says the mammalian target of rapamycin pathway promotes aerobic glycolysis and esophageal squamous cell carcinoma by upregulating pyruvate kinase M2 isoform. Both proteins can serve as a molecular target for novel therapeutic strategies. And I will say that I'm going to skip down here for a second. You can see here that the same old pathways, right, that we saw in other enzyme systems, whether it be hexokinase, whether it be pyruvate, phosphofructokinase, whether it be PFKFB, whatever. A lot of all of these enzymes are under the control of kind of the same kind of regulators. So here's ERK, here's PI3K, AKT, mTOR. We have PKM2 serving as a transcription factor, but we also have HIF1. We have NF-kappa B, or the inflammatory pathways. We have the beta-catenin. We have STAT3. My point by saying this, by showing all of these mechanisms, is that these mechanisms, the reason why they're scary, I should say, and the reason why they're not good, in particular with cancer, is because what it's doing to all of these enzymes is upregulating these enzymes, and therefore leading to the ultimate, effect, which is Warburg metabolism and the burning of glucose at a much higher rate. The next thing I wanted to talk about before we start hitting targets is that pyruvate kinase has roles in maintaining redox homeostasis. Okay, so this paper was published in May of 2024, just a year ago. It says exploring the diverse roles of PKM2, or pyruvate kinase M2, in cancer, navigating beyond glycolysis and the Warburg effect. So again, we've looked at beyond glycolysis when it's talking about transcription factors, but this is another distinct role that pyruvate kinase has. So it says here, pyruvate kinase M2, a key enzyme in glycolysis, has garnered significant attention in cancer research due to its pivotal role in the metabolic reprogramming of cancer cells. Originally identified for its association with the Warburg effect, PKM2 has emerged as a multifaceted player in cancer biology. The functioning of PKM2 has intricately regulated at multiple levels, including controlling the gene expression via various transcription factors and non-coding RNAs, as well as adding post-translational modifications that confer distinct functions of the protein. Here, we explored the diverse functions of PKM2, encompassing newly emerging roles in non-glycolytic metabolic regulation, immunomodulation, inflammation, DNA repair, and mRNA processing beyond its canonical role in glycolysis. The ever-expanding list of functions has recently grown to include roles in subcellular compartments, such as the mitochondria, extracellular milieu as well, all of which make PKM2 an attractive drug target for the pursuit of therapeutics in cancer. So the reason I picked this picture in particular was interesting because basically what they're showing here is that PKM2, when it's not oxidized, okay, when reactive oxygen species are not in excess, okay, PKM2 will, see if I can find a good graphic here. PKM2 will participate in converting phosphoenol pyruvate to pyruvate. So it'll make ATP for the cancer cells, and it'll ultimately lead in lactic acid production. Okay. So this is the Warburg metabolism, right? But it doesn't just do that. It also controls how much glutathione and antioxidant capacity the cancer cells have. So what it's saying here is that when, let's say high amounts of ROS, let's say we give you, I don't know, we give you radiation therapy, right? Or some chemotherapy that enhances reactive oxygen species, or some, I don't know, some effect of enhancing reactive oxygen species, like massive amounts. Well, what happens is, in normal physiology, is that ROS will basically lead to apoptosis. Ultimately, it will damage the mitochondria terminally, and it will lead to apoptosis. In cancer cells, however, PKM2 can help stabilize some of these pro-apoptotic proteins and lead to the blockade of apoptosis. That's one way that PKM2 blocks apoptosis and helps deal with reactive oxygen species. The other way, I think, is also extremely fascinating, and it's very similar to what we saw in the PFKFB, which I'm sure that you all remember like it was yesterday. But being a little sarcastic there, because I know I'm throwing around a lot of acronyms from enzymes that are very obscure. But my point is that we see that there are metabolic switches within this system that allows for the cancer cell to survive, right? So when ROS is present, or ROS, reactive oxygen species, oxidative stress is present, PKM2 gets oxidized. Then instead of glucose being shunted to lactate and making energy, okay, what happens is it will divert that glucose. That glucose will get diverted, and it'll go through the PPP, right? The PPP, the pentose phosphate pathway, is responsible for the generation of reducing products and the recycling of glutathione. So that way that the cancer cell can upregulate glutathione and other reactive oxygen species scavengers or antioxidants that can lead to ROS detoxification, and the tumor cell can survive, okay? This is what I talk about in a lot of videos, mostly when we're talking about glutamine as the internal shield system, okay? Now, these are like the inner workings of the internal shield system. You know, when we were talking about glutamine, we were talking about specifically the SLC7A11, or the XC transporter, which is a glutamine, or glutamate, I should say, cysteine antiporter, which helped bring in the extra cysteine necessary, and cysteines necessary to build new molecules of glutathione. Well, how is this contributing? Well, the glucose side of the house contributes substrate to the PPP, and the PPP is responsible for regenerating glutathione once it's made. So once glutathione is being used up, then it has to be regenerated by the PPP, by NADPH to be specific. NADPH is being made through the PPP, and therefore it helps detoxify, or maybe a better way is neutralize, the reactive oxygen species excess, which allows survival. The one way that it was put in one of the papers that I saw, it says that it puts the cancer cell in a theraptosis resistant state. Okay. So what this is doing is that PKM2 is one of the main switches, PKM2 as well as PFKFB, and I'm at 1163. I just want to like quickly show what I'm talking about here with PKFB. This is the slide I was talking about. So we see here that glycolysis, glucose is going to be shunted towards lactate, right? Here's our PKM2. What's going to happen is we have a metabolic switch here with PFKFB, excuse me, where it will shunt the glucose down towards lactate or back towards the PPP to have glutathione GSH redox homeostasis. Well, this is one of the major switches here, PFKFB. We talked about this in the past, but pyruvate kinase is the other switch, right? So basically, when glucose gets far enough down here that there's oxidative stress, it will reshunt the glucose back towards the PPP so that you can help maintain redox homeostasis and cancer cell survival. This is the internal shield system for cancer that we are trying to basically overcome with metabolic therapy. And it's a very elegant way of doing it, because what we're doing is we're effectively shutting down glucose utilization by cancer cells, right? So now we don't have the energy to make we don't have the energy production from ATP. We don't have the PPP substrates to recycle glutathione and also, proliferate and maintain redox homeostasis. And then because glutathione is a third of it is glutamine, right? Or glutamate, it has to be converted to glutamate. Now we can't make glutathione. Now, when we restrict glutamine and glucose, we are leaving the cancer cell vulnerable because energy is not being produced through substrate level phosphorylation or basically energy produced by glucose and glutamine. And these cancer cells can't make or recycle glutathione. So they are very vulnerable to redox imbalance. Okay, here's the yin and yang, right? We're gonna leave it in a theraptosis susceptible state. And then the third leg of the stool of the way that metabolic therapy is laid out is that you then hit the cancer cell with oxidative stressors, preferably oxidative stressors that don't destroy the normal tissue, like radiation therapy or chemotherapy, but more like IV vitamin C or HBOT or ozone therapy or soup du jour, maybe photodynamic therapy, etc. We upset this redox homeostasis on purpose when it's in a maimed state, so that we lead to the ferroptosis and cancer cell death that we're looking for. That's how it works. So we're seeing here that, and if I can find my space here, I've got myself off kilter here. But my point is if we can see that pyruvate kinase M2 is a key component to this redox homeostasis and this kind of internal shield system, we can see that maybe pyruvate kinase M2 could be an effective target. Now, before I go any further, I'm going to give one more example of what it can do. It can also affect tumor immunity and build new blood vessels. Okay, so this PKM2 is actually being transported outside the cell, and it helps contribute to the building of new blood vessels called angiogenesis. It will then autocrine fashion. It will then stimulate the EGFR receptor in a vicious cycle, even a further vicious cycle, and then it will lead to immune modulation and dysfunction, so that it will help kind of hurt our immune system from being able to attack the cancer cells. So it's got a bunch of different things that make it a pretty attractive target. Now, before we go into actually specific things that can block it, I want to show this list, okay? So the prognostic significance of PKM2 in human cancers, okay? So this is a huge list of different cancers that if PKM2 is increased, the prognosis is poor, okay? So you see poor, all the way down, right? Every cancer that it's been studied in, if it's increased the amount of PKM2, poor prognosis, okay? Which makes total sense if you've been following the channel, because you know that the further along we are from using oxidative metabolism to Warburg metabolism, the more malignant and aggressive it is. It doesn't matter what kind of cancer it is. And therefore, it would make total sense that the more PKM2 is available, the poorer the prognosis would be, okay? But this is, mesothelioma, gastric cancer, cholangiocarcinoma or gallbladder cancer, melanoma, ovarian cancer, head and neck cancers, esophageal cancer, liver cancers. I mean, you name it, right? Pancreatic cancer, basically every type of cancer. So my point is that so you may say, well, I have X cancer. Is this going to be important for me? Very likely, yes. Not only because of this graphic, but because we also know that, enhanced glucose utilization, Warburg metabolism, mitochondrial substrate level phosphorylation are hallmarks of cancer, all cancer, including non-solid cancers as well. We talked about this slide here, how different signaling pathways all lead to Rome here. They all lead to PKM2 and other mitochondrial enzymes. I don't want to like make it seem like it's only PKM2 in a vacuum. It's all of the metabolic machinery necessary to upregulate the use of glucose to, burn sugar for energy and for biosynthesis. And so, but you see the same players over and over again, the same metabolic contributors. And so let's go into how maybe the use or the inhibition of PKM2 could enhance some standard therapies first. So this paper was published in December of 2018. It says enhancing 5-fluorouracil efficacy through suppression of PKM2 in colorectal cancer cells. It says here suppression of PKM2 resulted in changes in glucose metabolism, of course, leading to decreased synthesis of ATP. Makes sense. Reduced ATP to ADP ratio, which resulted in the intracellular accumulation of 5-FU or 5-fluorouracil, consequently enhancing the therapeutic efficacy of the drug in several colorectal cancer cell lines. We showed the combination treatment showed superior anti-cancer efficacy compared to 5-FU alone. These findings suggest that targeting PKM2 can increase the efficacy of chemotherapy, potentially providing a new approach for improving the outcomes with chemotherapy in patients with colorectal cancer. So, as with most things we've talked about with ketogenic metabolic therapy, and really like anything that enhances metabolic therapy is very likely going to enhance the standard of care as well. Okay, so this is just one example of it enhancing chemotherapeutic regimens. It will also enhance radiation therapy as well. And this paper was published in November of 2023. And it says M2 isoform of pyruvate kinase rewires glucose metabolism during radiation therapy to promote an antioxidant response and create glioblastoma radioresistance. So it says here, we showed that radiation therapy induces glucose utilization, increased utilization of glucose in GBM cells. So this is one of the first papers that I've actually seen that showed that this does this. But this is what Dr. Seyfried kind of talks about on a lot of his podcasts. He says that when radiation therapy is employed, it will upregulate the use of glucose. And it's saying that it will liberate glucose. But in this paper, it's saying that it'll actually induce the utilization of glucose by GBM cells or glioblastoma or primary brain cancer cells. And this is accompanied by the translocation of GLUT3 transporters to the cell membrane. So basically what they're saying is radiation therapy is going to upregulate glucose transporters to the cell so that glucose can be utilized by these GBM cells more readily. That's a problem. An irradiated GBM cell routes glucose carbons through the pentose phosphate pathway, the PPP, to harness the antioxidant power of the PPP and support cell survival after radiation. These are the internal shields we're talking about. So when the internal shields of the PPP and the NADPH and glutathione, that actually increases not sensitivity. The word I want to use is resistance to radiation therapy. Okay, which is why, for example, very likely that like HBOT or IV vitamin C in a vacuum is probably going to have little or not the effect you're looking for because of these mechanisms. Okay, this is why radiation therapy fails. This is why IV vitamin C, which makes total sense by itself, is not going to be the magic bullet. This is why HBOT is not going to do a lot for yourself for cancer unless you hit the other legs of the stool, and glucose being one of those, right? This response is regulated in part by PKM2. Activators of PKM2 can antagonize the radiation-induced rewiring of glucose metabolism and radiosensitized GBM. So what they're saying, and I think it's a very fascinating take on this, is what they're saying is that we talked about in this slide here that when reactive oxygen species, what's happening is it will oxidize PKM2, inactivate it effectively, and it will shunt glucose towards the PPP. And that way it will deal with reactive oxygen species. What these researchers are saying is that if they make the inactive PKM2 that was hit by reactive oxygen species active again, it will allow the cancer cells to be a little bit more radio resistant, because now the PPP is less active. There's less glutathione regeneration, and the reactive oxygen species generated by radiation are then active again. But the downside to that is that when PKM2 is active, it's going to then make more lactate, which will then create its own next vicious cycle. So I don't think that's the real answer, is to just activate PKM2. That's how, I mean, I think it's interesting what they're saying, but it doesn't make sense from like a long-term kind of like holistic perspective. Why would we want to like allow more lactate? Lactate is part of the reason why your immune system doesn't work. Lactate is part of the reason why you have the vicious cycles going on inside the cells, et cetera, et cetera. So therefore, I just don't think that's the right strategy, but it's an interesting take on what this is saying here, and a way to enhance radiation therapy. I think the goal shouldn't be to enhance radiation therapy. The goal should be to help kill cancer cells and preserve our normal brain tissue, right? Like, I don't want to have to radiate somebody's brain. So like, although that's an interesting take on how to enhance radiation therapy, and it makes a lot of sense, it also probably is a very short-sighted way of looking at this. Radiation increases glucose uptake in these gliospheres via increased membrane localization of GLUT3. Radiation can also suppress PKM2 activity, leading to a bottleneck and glycolytic flux accumulating of glycolytic intermediates, promoting flux to the PPP and the generation of antioxidants, which have the potential to neutralize the oxidative stress induced by radiation therapy. Thus, the use of PKM2 activators can prevent the suppression of PKM2 activity by radiation, thus antagonizing the antioxidant function of PPP. Again, I think that's going to be an interesting take on it. I think it's the wrong strategy, but that's one person's opinion on this. Let's see here. So I'm going to quickly go through this part of it. Okay, this is the part that I would call this maybe the climax of the talk, or even the little micro series on PKM2, is that we have a lot of tools in our tool bag of things that can actually affect PKM2. Unlike a couple of the more kind of smaller enzymes, such as PFK, PFKFB, there was not very much literature to show that, you know, these things are having an effect. Whereas we have a ton of things. I went through the literature further and, you know, found some key things that could have effects here. You know, there's a bunch of graphics like this where there's things that can, affect it. Here's another one here. I've circled these with red, showing that it has effects on PKM2. Lots of graphics like that, but I wanted to show a little bit more detail here. So, quercetin was one of my favorites of all time because it has so many unbelievable things. Well, quercetin, if you look at it, is dose and time dependently able to reduce the extracellular acidification rate as a measurement of glycolysis in these melanoma cells, inhibited. And one of the ways that it did it was it inhibited glycolysis at the level of the PKM2 enzyme, a key control point of glycolysis, by 50% at a level of 9.24 micromoles. Moreover, quercetin decreased the protein level of expression of GLUT1 and breast cancer. We know about that. So I just wanted to give you some examples here. So they use in these studies, these are, I believe, rodent studies, they were using 50 milligrams per kilogram twice a month, which reduced the PKM2 levels in tumor tissues of breast xenograft models. That indicated that quercetin may reduce glycolysis by targeting key enzymes of glucose metabolism and uptake. Interestingly, the quercetin analog, isoquercetin, increased the activity of recombinant PKM2 by 50% at a concentration of 1.34 micromoles. So what they're saying is that it took 9 micromoles of concentration of quercetin to lower the amount of this PKM2 by 50%. However, if they use this analog isoquercetin, it lowered it by 50% with like nearly a 9-fold decreased level of concentration. So it just shows that it's much stronger at decreasing that molecule. Apigenin, another kind of like molecule that I have not had a chance much to talk about, but I wanted to give it its day here. Apigenin restrains colon cancer cell proliferation via targeting blocking of pyruvate kinase M2 dependent glycolysis. What I liked about Apigenin in particular is that it had roles at actually blocking the enzyme directly and lowering its expression also. So it says here, we found that Apigenin could block cellular glycolysis through restraining the tumor-specific pyruvate kinase M2 activity and expression, and further significantly induce anti-colon cancer effects. Interestingly, the presence of PFB, an inhibitory effect on AP, the inhibitory effect of, let me slow down here for a second. Interestingly, the presence of D-fructose 1,6-diphosphate, PFB, the inhibitory effect of Apigenin on PKM2 was not reversed. So that's a very strong activator, and it was not reversed when in the presence of AP, of Apigenin. It said that AP could ensure a low PKM2, PKM1 ratio, thus blocking its further signaling with beta-catenin pathway. Ultimately, it led to arrest of colon cancer cells through directly affecting PKM2 activity and expression. There's another paper here kind of showing a very similar thing. And ultimately, we've already gone an hour already, haven't we? We've already done 48 minutes. Let's see here. Tannic acid also had a direct effect on PKM2. Beta-Elemene. Beta-Elemene was really strong at doing this. And beta-Elemene is found in white curcumin, curcumin xylitol. And there's a bunch of really neat graphics showing PKM2 being completely blocked by beta-Elemene. So let's get to the last slide here, maybe the slide that most of us have been waiting for. So this is everything that I could find that could inhibit or downregulate pyruvate kinase M2. Some of these things we've seen multiple times in the past: artemisinin, dihydroartemisinin, metformin, genosinicide, apigenin, quercetin, resveratrol, curcumin, white curcumin in the form of beta-Elemene, EGCG, sulforaphane, silibinin or milk thistle, ellagic acid. Multiple things have an effect here. We talked about ivermectin in the most recent video that I actually published, not a live stream, the video. And then K3 and K5, I saw this K3, K5 activity. It's just that those are like artificial, what I can see, artificial K vitamins that are kind of given mostly in animals. So I don't think we're going to be using those. But my point is, by showing this, is that we have a ton of tools in our tool bag. And I hope this was helpful. If you like videos like this, please like. If you have people in your life who have cancer that we know are going to be running off glycolysis and upregulating glucose and the use of glutamine, and they need help, please point them this way. And until next time, guys, I'm sorry. I got to cut off a little bit early today just because it's already been an hour and I have another engagement that I have to attend to. But it's been a pleasure. And if you like videos like this, please subscribe if you're not a subscriber, and share it with those who you love and care about, who could potentially use this information. And until next time.